Hi everyone, I'm Menghan. So today I'd like to discuss about the nucleic acid. So nucleic acid is a polynucleotide. So basically, the monomers of the nucleic acid is the nucleotide. So nucleotide has three components. It's called the pentose sugar, means has 5 sides, nitrogenous groups, and phosphate group. So nucleic acid is divided into two: deoxyribonucleic acid (DNA) and ribonucleic acid (RNA). So for the pentose sugar, we have ribose sugar that is in the DNA and RNA, deoxyribose sugar in the DNA. So what is the difference between the two pentose sugar? The difference is at the second carbon atoms, that is the RNA, have extra oxygen atom compared to the deoxyribose sugar. Okay, so for the nitrogenous groups, it's based on the nitrogen, based on the number of the carbon rings. So they have single rings called the pyrimidine, and also double rings called the purine. So pyrimidine has 3 types. There is the cytosine, or called C. Uracil only occur in the RNA, and thymine only occur in the DNA. And what for other purine? Purine has 2 types. Adenine and guanine occur in both DNA and RNA. So phosphate group, the chemical formula is PO4. PO4. Yeah, PO4. They has OH minus, OH groups. So nitrogenous base in the DNA has guanine, adenine, thymine, and cytosine, and for the RNA has guanine, adenine, uracil, and cytosine. Okay, so for the DNA groups, DNA nucleotide, the phosphate group is on the left hand side. They will connected to the carbon number 5 at the deoxyribose sugar, and the deoxyribose sugar will connected to the nitrogenous base through the carbon atoms number 1. This is the arrangement of the DNA nucleotide. And RNA is also the same, but the difference is the nitrogenous base for the RNA is double sugar. Nitrogen base is the RNA is double sugar. So formation of the dinucleotide. Dinucleotide means 2 nucleotides joined together by condensation process. So you will produce the H2O. So for the H2O formation, the OH minus group is removed from the OH group from in the phosphate group of one of the nucleotide in the DNA, and another is for the H group at the 3rd carbon of the pentose sugar of another molecule of nucleotide. So joined together by phosphodiester leakage or phosphodiester bond. So for polynucleotide means many nucleotides are joined together by condensation process, and the elongation is from the 5 prime, means the number 5 carbon atoms to the number 3 carbon atoms. Yeah, and and for the others is the DNA structure based on the Watson and Crick's model, and they found that the guanine, and in the DNA, the guanine and the cytosine will join together, and the adenine and thymine will join together. Yeah, these they are found. So for the thymine and adenine, they are joined together by 2 hydrogen bond, and for the guanine and cytosine are joined together by 3 hydrogen bond. So for RNA has the single strand only, so means he has a single polynucleotide, single strand of polynucleotide, so has 3 types: messenger RNA, rDNA, and tRNA. So they has their function respectively, and will be details in other chapters. So the difference between DNA and RNA, so most of it has been discussed. For other properties, like DNA is susceptible to the UV radiation, DNA is weak weak to their UV damage. For RNA is relatively resistant to the UV damage. And for DNA, because it's double strand, so he has a larger molecular size. And for the RNA, he has a single strand, so he has a smaller molecular mass. So that's for me. Thank you.